concerns of damaging side effects of traditional cancer treatments, such as potent chemo drugs and radiation, there's been a recent reshift in focus in the cancer research world. Cancer scientists are now interested in whether cures can be found not externally, but rather from within, inside the bodies of each and every one of us. For my research on ovarian cancer, I am studying how to enhance the abilities of an immune cell that we all possess, called the killer T cell, which is able to act as a soldier within us to infiltrate tumor and induce cancer cells to die. This immunotherapeutic approach could open the doors for much needed alternative treatments for ovarian cancer, which remains the leading cause of death from gynecologic cancer in the U.S. Ovarian cancer is known as a silent killer because it's very challenging to diagnose early, and it has a very dismal 5-year survival rate of around 25% because of its inability to be diagnosed early, compounded by issues such as metastasis and rapid development of resistance to first-line chemo drugs. Excitingly, it was found that more T-cell infiltration in tumors is significantly correlated with improved patient survival. Though this observation is great, making a therapy that capitalizes upon this observation is not as simple as it seems. Unfortunately, tumors are smart, and they developed ways in which they can evade normal immune response. As you can see in this picture, tumors are not simple aggregates of malignant cells, but they're rather pretty dynamic bodies to which they can recruit and corrupt surrounding cells and establish this immune suppressive network that prohibits T cells from coming in and killing off tumor cells. So for my project, I focused in on 2 specific components of this tumor microenvironment. I was interested in studying tumor cells themselves, as well as tumor-associated blood vessel cells, and understanding how these 2 different components contribute to immunosuppression. So my project can be broken down into 3 very specific aims. Firstly, to identify novel immunosuppressive genes expressed in tumor cells and tumor-associated blood vessel cells. 2, to determine the mechanisms by which these genes link to poor T-cell infiltration to tumors. And finally, the ultimate goal would be to develop gene silencing nanotherapy that can basically knock down this gene and associated pathways to improve T-cell infiltration and ultimately improve patient survival. So to start off, I first looked at 120 ovarian cancer patient tumors and identified this one gene, and I'll refer to as gene X for patent-related purposes, that was significantly correlated with poor T-cell infiltration. Next, when I looked at patient data, I found that high gene X expression is strongly correlated with worse patient survival. So this data strongly supports gene X as a novel candidate for our therapy to come and shut it down. From extensive cell experiments, I found that gene X actually acts as a master regulator to control various immune suppressive pathways. As you see in the image in the green here, gene X controls and manipulates several different genetic pathways that lead to poor T-cell infiltration to tumors. So to sum up my project, I found an novel immunosuppressive gene called gene X, which is associated with poor T-cell infiltration to tumors and poor patient survival. Secondly, my work established specific mechanistic ways linking gene X to poor T-cell infiltration. And finally, the greater goal of the project, which I've been contributing to as a whole, is the development of a novel nanoparticle therapy that can come in, target tumor, and deliver gene silencing technology to shut down gene X, improve T-cell survival, and ultimately improve T-cell infiltration, and ultimately improve patient survival. Thank you for listening.